Hello, my name is Daniela Dulworth, and I'm a senior here at Case Western in the biology department. And my research studied the genome variation in segregating populations of flax. And so a little background on flax: it is a stable crop, and there are two main lines of flax that we use. It's Bethune and Stormont Cirrus. And Bethune is mostly used for its oil-bearing properties, and Stormont Cirrus is used for its ability to produce fiber used in linen. And Stormont Cirrus is also known as PL, that's what I'll call it, and it is able to respond to environmental stressors. So specifically in low nutrient conditions, there are certain genetic events that occur that allow it to grow better in these stressful environmental conditions. The main genetic event that occurs is the insertion of Linum insertion sequence 1, or I'll call it LIS1. And this is very important, and it's the main thing that I studied this semester. So LIS1 is a 5.7 kilobase insertion into the PL genome in flax, and this happens under low nutrient conditions. So when the insertion occurs, flax is able to grow better in stressful conditions. It is a heritable and site-specific induction. So what that means is it's only going to happen in one specific place of the genome, and any progeny of the plant that has been induced will also have the LIS1 insertion. And there are a couple of theories of how LIS1 is induced and made, but the main idea right now is that there are fragments of DNA assembled outside of the genome and then inserted in when the inducing conditions occur. Some people believe that it might be like a transposon, but due to the makeup of LIS1, that is not the accepted theory. And so what picture we're looking at right here shows the insertion sequence, and the three main primer pairs that we made are based off of basically this photo, and we look at the flanking regions of the insertion sequence. So if you look at the 2 and the 3 prime, that's the left-hand flanking sequence, and we study that, as well as the 18 and the 19 prime sequence. And then to tell if there was no insertion, you look at the 2 and the PL9, because that's the uninduced sequence. That's where it's actually inserted into on the flax genome. So in our methods, what we did was strains of PL and Bethune were grown up in non-inducing conditions, and then they were crossed. And this cross, the F1 generation, was also grown up in non-inducing conditions. And the progeny of one of these crosses, we call family 35, was grown up in inducing conditions. And we studied this family. There were about 51 samples to see whether or not LIS1 was inserted and if genetic changes were occurring. Then we collected leaves and meristems from the tops of these plants and extracted DNA from them. We ran PCR in them and then ran them in agarose gels to determine if bands were present and whatnot. So the 5 main primers we used were the 2 and 3 prime, 18A and 19 prime, and the 2 and PL9, which specifically tell us if LIS1 was inserted. And then we also use 2 scaffolds. Scaffold 1 is on the 11th chromosome, and scaffold 2 targets the 7th chromosome, or scaffold 4 targets the 7th chromosome. And these scaffolds allow us to know not necessarily if LIS1 is being inserted, but if genetic events are occurring in response to the stress conditions. So here are the results from the 3 primers that tell us about the insertion, and they're just a bunch of gels. We'll get into it later. And here are the results from the scaffolds. So looking at table 4, that's actually tells us about the insertion of LIS1. You see 2, 3 prime, 2 PL9, and 18 and 19 prime. And you also see an induced row, and that column will show you whether or not the LIS1 has been inserted. A plus sign means it has, and a minus sign means it hasn't. And there are about 18 blank spaces, which means we got nothing from the information. So you can see also in some of the samples that there is 2 PL9 2, 3 prime, or 18A, 19 prime. Basically, what 2 and 3 prime and 18A, 19 prime tell you is that there was an insertion. And sometimes there's only one or the other. That means that it's only started to be inserted on either the left-hand flanking region, which is 2 and 3 prime, or the right-hand flanking region, which is 18A and 19 prime. The presence of a 2PL9 band means that there is no insertion, but the reason that you might see all three is because that leaves are collected, and if the plant at the top had been induced, but leaves below had not been induced yet, and all of those leaves had been collected, you might be seeing a homogeneous mixture of all of this. And looking at table 5, this is breaking down our scaffolds. So there are, looking at scaffold 1 first, which is that 1 LNR, there are 3 major band sizes that we're seeing, and they are 750, 1000, and 850. And basically what that's going to tell us is that on the 11th chromosome, what type of genetic information we're seeing. 850 means that it is just inherited genetic information from the Bethune parent. 750 is telling us that it's inherited genetic information from the PL parent. But 1000 base pairs is going to tell us that the genetic changes are occurring, and that not necessarily that LAS1 is being inserted, but something is changing and that the plant is being induced. And so you might see a mixture of 750 and 1000. That it just means the same thing as if you'll see all 3 of the 2, 3 prime, 2PL9, and 18 and 19 prime, that multiple leaves were collected. But as long as you see 1000, it means it's being induced. And similarly to the scaffold 4, there are 3 major band lengths we're seeing: 500, 1000, and 1500. So 500 is from the PL parent, but then also 1500 and 500 are from the Bethune parent. So that lets us know that 500 is shared genetic information from both parents. But then the 1000 base pair length is also going to tell us that there is induction occurring. And the same thing, if there's 1000 and 500, it's still induction. And so in conclusion, there were 29 samples that had the LIS1 insertion, and 4 of them had no insertion, and 18 of them lacked any indication of insertion based on table 4. But what you can do is, looking back at table 4 and table 5, you can cross the information. And for 4 of those samples that had no insertion, they actually are showing that there is some genetic changes occurring from scaffold 1 and scaffold 4 due to the 1000 base pair length appearing in their gels. And when you look at those 18 samples that were lacking anything in the first 3 primers, when you compare them to the scaffolds, only 3 of them actually show that there was genetic events occurring and that induction was occurring. And so in the future, we can look at the whole genome of all of these samples, and that is what is supposed to occur next semester, along with some Sanger sequencing, in order to determine what the differences are and why we're having inconsistencies among these 18 samples that are lacking the first 3 primer pair bands. Thank you.